So I started in the field of genetic engineering when I actually started my PhD. So I chose the lab because of its expertise in genetic engineering. And back then I already knew that I wanted to work in that field. So I started to use CRISPR when I actually came back from a conference in what is now a very famous conference in Italy, 2012, where everything was about, back then, talents and zinc fingers. And the very last talk was about this CRISPR. And so everyone was very excited about it, so was I. So I went back to the lab and ordered the first Cas9 expression plasmid and guide RNA expression plasmid available from Agene and started to do my experiments. So I think the biggest achievement I had with CRISPR was actually doing my first experiment with it. So back then I was trying to engineer mouse embryonic stem cells and to basically endogenously tag genes of interest with fluorescent markers. So I designed guide RNA to the locus of interest and then used CRISPR along with a donor molecule to achieve the tagging, the targeting. And to my surprise, we got 90% targeting efficiencies, and strikingly, we even achieved B allelic targeting at 5%, and this is something we never achieved before. So for the first time we were able to tag a gene at both alleles at the same time. So there's no doubt that CRISPR is and has changed the way we do experiments, and it's a very exciting times to work in that field, but not only in biology. I think it's affecting all other life sciences as well. And I think we're going to see some exciting results coming from the clinic in the field of immuno-oncology in the near future, but also in the field of agriculture, where we now are able to engineer crops. So the main challenges for CRISPR are still to achieve large knock-ins. So we can use CRISPR to produce a double-strand break at a very precise location, and we can harness this to generate knockouts and small knock-ins. But one of the main challenges still is to achieve larger knock-ins. There's also the problem of how to deliver, for instance, in a clinical application, the reagents to the right cell. That has not been solved with CRISPR. And I think personally, I think the biggest challenge is actually to educate and inform the people out there, the non-scientists, about this new technology. So my top tips would be keep it simple. The most challenging part of a CRISPR experiment itself is not the transfection anymore, it's about more the design. So what I mean by this is have a good strategy in place, design a good guide RNA, and also make sure that you have a good genotyping strategy in place. You don't want to end up with a lot of pups or clones and have no means of screening for them for the mutant.